Hello, welcome to Plant Party, Getting to the Root of Soils, hosted by Texas Parks and Wildlife Department, Texas A&M, AgriLife Extension Service, the USDA Natural Resources Conservation Service. Plant Party is a quarterly webinar series intended for advanced plant training and fun education. I'm Megan Clayton, professor and extension range specialist with Texas A&M AgriLife Extension Service. My co-hosts are Tim Sigmund, the private lands program leader with Texas Parks and Wildlife Department, and Charles Kniper, state resource conservationist with USDA Natural Resources Conservation Service. We have 15 wonderful door prizes we'll be giving away today as a part of today's event. Our door prize donors today are committed to supporting you in your quest to learn more about plants. I'd like to thank Bamert Seed, InView Range and Pasture, Native American Seed, Texas Quail Forever, Renewable Resources Extension Act, Texas A&M AgriLife Extension Service, and the USDA Natural Resource Conservation Service for donating these neat prizes. Our first presenter today is Lee Knox. Lee was with USDA NRCS for over 20 years before recently taking a role with Texas Christian University as a professor in ranch management. Prior to NRCS, he worked as a county extension agent for Texas A&M AgriLife Extension. So you can see he has extensive knowledge of managing rangelands, and we're happy to have him kicking off our plant party today. Lee received a BS in range from Texas A&M University and a master's also in range from New Mexico State University. Today, he'll be presenting on rangeland soil health. Thanks for starting us off, Professor Knox. I'll turn it over to you.
Thank you, Megan. I appreciate the opportunity to visit. A little side note real quick about the TCU Ranch Management Program. We've been around since 1956. Currently, our graduates manage in excess millions of acres of rangeland across Texas, Oklahoma, Kansas, and New Mexico. And up in Montana now. So anyways, we've been around for a while, and it's my job to help with their education on rangelands. So let's define rangeland soil health. We got to start with a definition of rangelands, lands on which the indigenous vegetation, whether it's climax or the natural potential, is predominantly grasses, grass-like plants, forbs, or shrubs, and is managed as a natural ecosystem. If plants are introduced, which is very common, you can see that in the bottom of this picture, there's KR bluestem in front of the little bluestem. These introduced plants are managed similarly, so with natural systems. Rangelands include natural grasslands, savannas, shrublands, many deserts, tundras, alpines, communities, marshes, and meadows. If you want a biblical reference, a good biblical reference is anytime the Bible says they went out into the wilderness, essentially they went out to rangelands. And so rangelands are a lot of the forsaken country, as we say. A little other thing about rangelands is they developed under conditions of periodic disturbance. They are a disturbance-driven ecosystem. Those disturbances include drought, picture to the far upper left, fire, the wildfire in the right picture, and the fire prescribed burn in the bottom right picture. Grazing by domestic animals and native wildlife, weed incursions, which we'll see some pictures of here in a little while, outbreaks of insect pests. Not only is that a drought in the top left corner, that is subterranean termite infestation also on top of drought, creating all that bare ground. Plant diseases and human activities all impact rangelands, but they are a disturbance-driven ecosystem. So let's talk about soils and basic. Now, I apologize, I have a pretty thick draw, so soils is very difficult for me to say, so I'll probably mess it up somewhere during this presentation, it'll come out as soil. So anyways, the major components that we all think about it as a mineral part, but the mineral component only makes up 45%. 25% is air and 25% is water. And when it comes to soil health, the most important component is that organics part, which is roughly 5%, and that will vary depending on what part of the state of Texas you're in. But primarily, if typical soil will say 5% organic matter. And so from that, you can see there's organisms. They're only 5% of that 5%. Living roots is 10% of that. And there's that humus. And that's not hummus. That's not fancy bean dip. That's humus. And so humus is long-term organic matter. And the neat thing about humus is what it does, it is it has the water holding capacity 4 times greater than clays, and it is the most reactive component in soils. So that 5% is the most reactive component in soils, even way more reactive than clay particles. So the number one thing when it comes to soil health, number one by far, primo, top of the list, everything you need to think about is keep your soil. Erosion is an issue. It continues to be an issue. And it doesn't matter how healthy it is, if it's on the neighbors, it's no longer yours. And so keep your soil. So we have a picture of wind erosion in the top. We have a gully erosion occurring in the middle picture. The one that most people don't see or they step right over is water erosion. And what you have there is what we call a terracet. And that is water has moved that soil downhill. It's hit that little bit of a obstruction of vegetation and caused a terrace. And so that is the active movement or erosion of soil. So the number one thing is we can't make it better if we can't keep it where it's at. A little shout out here. The 6 points I have on soil health, rangeland soil health, come from a little publication by Texas GLC and the USDA NRCS, Soil Health Principles for Profitable Ranching. So that's the basis of the rest of the presentation today. And then from there, I'll add a little bit of additional context. So number one, know your context. Know where you're at. So I have the Gould's ecoregions of Texas. When it comes to soil health, there's some rules of thumb that we can abide by. And so in regions 1, 2, 3, and 4, we have tremendous amount of rainfall compared to the rest of the state, and we can grow a tremendous amount of biomass. And that biomass turns into soil organic matter in the soil later on. So we can grow a whole lot and it doesn't rot as fast as other parts of the state. And so because of that, we can actually have a really fast impact on soil health. When you go down into Region 6 in the South Texas, they can grow a significant amount of biomass down there, but because it never freezes or rarely freezes down there, soil microbes are burning that organic matter up as fast as we can grow it. So it's really more difficult to grow additional or to accumulate organic matter in the soil when you get down into far South Texas. We'll skip over and go to regions 9 and 10. Rainfall amounts out there, doesn't rain a lot. And so pretty well 12 inches, 14 inches or less in regions 9 and 10. And because of that, you don't grow a lot of biomass. If you're not growing a lot because it's moisture limited, you don't have the potential to accumulate a lot of organic matter. So it makes it more difficult in those environments to really have an impact on soil health or a fast impact on soil health. Now, when you get into regions 5, 7, and 8, and that's where I spent a majority of my life and my professional life, when you get in those regions, it's more difficult. We can see a change, but it's over a longer period of time. And so they are kind of the edge of the world, the edge of the growth potential. And because of that, we can make an impact, but the impact, you don't see it. And it's not a year or two event. It takes 3, 4, 5, up to 10 years to really have an impact on soil health in those regions. So principle number 2, cover the soil. And so my I have this picture here split. I love fence line contrast. So the pasture on the right has a really good coverage, has a lot, everything is covered well, has big growthy plants on it. And the pasture on the left is grazed rather hard, more bare ground, more open space. The ground's not as covered as well. So the top part of this slide is from 1954. So we've known the principles of soil health, we just didn't call it that back then. And so on the left, with 1,700 pounds of forage production on the left, you can only store about 4 inches of rain in a 2-hour event. Whereas on the right, you can store 10 inches. Soil temperature is only 95 degrees on the right, and soil temperature on the left is 135 degrees. So just keeping that ground covered keeps the ground cooler, less evaporation, but most importantly, it allows us to absorb as much moisture as possible, especially when you go back to that previous slide, that region in that central part of the state, when you go through the central part of the state, we're still moisture limited, and so we want to capture as much as possible. The third, optimize disturbance. So we talked about the disturbances of rangeland. I have 2 pictures here. The picture on the left is grazing disturbance. And I don't know if you can notice it, but there's some light pale blue grass plants on that left side. That is a little blue stem. So the decision, this is some country I manage, I graze, I lease out, and I have the ability to run livestock on. And so we rotated out of this pasture because all of the other grass was going dormant. The little blue stem, because of its deep roots, was still growing. And so we rotated those out so they don't start overgrazing the little blue stem, because that'd be the only green species left, forage left in that pasture. The other one is a burn we did, 2 burns we did. On the right was a spot out on a summer burn. And so you can see the forward response the following year. That forward response is from a significant amount of bare ground. On the left side of that annual broomweed line is the response from the winter burn that we burned it later, and we burned that in February. And then we have a much more, a better ground, not near as intense a fire, whole lot more litter, grass responded a whole lot better. And so we need to manage our disturbances well when it comes to our rangelands. I love this picture here. Increased diversity. If you look there in that picture, we have trees, we have shrubs, we have Texas persimmon, live oak, we have yellow Indian grass, probably Neely grama growing in, which is one seed grama growing in, it looks like side oaks grama, Texas wintergrass, vine mesquite, upright prairie coneflower, and one of those yellow asters, annual asters. If I tell you a name, it'll be wrong. And so you can see the diversity in this picture. In this picture, this is not a excluded range site. This is actually on the actively grazed, well-managed ranch in the hill country, and that runs cattle, sheep, and goats. So the fifth principle is to integrate livestock. And so the only thing I want to say about integrating livestock is, if you're going to integrate livestock, let's do it right. And so the picture on the left, not doing it right. You can see why I love these fence line contrasts. Look at all the bare ground. Whenever it gets hot in that picture, this picture was taken probably in February or March, probably March. When it gets hot in August with that much bare ground, that country on the left is going to roast. The picture on the right, you can see that mesquite forest. We would like to make that better, but at least they're doing the grazing correctly. And so, so we have this, principle of, you've always heard take half, leave half. And so how you graze impacts the root biomass, which impacts soil health below. So integrating livestock does help with nutrient cycle, makes it faster, makes soil health better. But if it's done incorrectly, it can be to the detriment very fast. Here's another fence line contrast in the hill country. You can see the place on the right, which is actively grazed. It's a cattle, sheep, and goat operation. Much better job of grazing management than the place on the left, which is also a cattle, sheep, or goat operation on the left. So it's all about the management. So integrating livestock correctly will help soil health. So this slide here, I love this picture. I got to take this picture on some lease country I have in the Edwards Plateau, South Taylor County. And I love this picture because you can see there is warm season forbs, cool season forbs, cool season annual forbs, cool season grasses. And if you look right down here in the center bottom, that's a little blue stem plant that's just now going to get started going. This picture was taken about the second week of May. And so that, by the way, that plant in the middle, that's a bottlebrush squirrel tail, which is kind of a unique plant to the region. I put the slide on the root growth. You want actively growing roots of all species, cool season, warm season, perennial, annual, everything growing. But the other one is when we harvest those roots or harvest those plants for grazing, if, we've always heard take half, leave half, but if you only take a third, the neat thing is that plant can continue to grow roots and it doesn't even stop, and it's just like it's ungrazed. So maybe instead of take half, leave half, we need to just say, let's just take a third. I'm going to end with that. One thing I want to say real quick about rangelands is it is the only land use that can produce a commodity: beef, lamb, mutton, cabrito, wool, wildlife species. It can do all those and can be managed with complete natural systems and can be done year after year after year if it's done correctly without degrading the resources. Really unique land resource, which I love. Thanks for the opportunity today, and I'll leave from there. Thank you.
All right, thanks. Thank you, Lee. Thanks for that great presentation. We're going to go right into our next speakers. Our second presentation today will be from Ashley Anderson, an incredible soil scientist with NRCS located in Temple, Texas. She has worked for NRCS for 15 years, starting as a summer intern while attending Texas A&M. And now she has spent most of her career in Texas with a little time in Arkansas, focused today on web soil survey. So Ashley, we'll pass it over to you.
Good morning, everybody. I'm going to do a quick web soil survey kind of demo run through and try to hit on a couple things that I feel like would be most useful for everyone here and more rangeland kind of focused. So we're starting here at the Web Soil Survey homepage. If you just type Web Soil Survey into whatever search engine you typically use, a lot of us, just Google stuff. So if you type that into Google, it should be one of the top links that comes up as a result. So when you get to this page, you just hit Start Web Soil Survey, and it opens another tab and brings you to this map of the United States. So you have to identify an area of interest. So you have a couple options that you can use. If it's an address, you can type in here. You can narrow it down by state and county, soil survey area, which typically follows county lines. There are some dual county soil surveys, so it would just take you to those two counties. If you have a GPS point, you can enter it here, and you even have, if you have a shapefile, you can import it through this way, but probably one of the most common ways is one of these lower ones. So I'm just going to do soil survey area, and since I work in Temple, we'll do Bell County. And if you hit you hit view, then it will zoom into Bell County. So from here within the map, you can pan around, and you can also then select an area of interest using either of these two buttons. This one makes you use a rectangle. This one lets you draw it out by just clicking around. So we'll just make a little area. Okay, and then when you double click, it will open again, and you will now have this new map with the diagonal turquoise lines. this is going to be your area of interest. So if you sometimes when you do it along the property line, you maybe want to give yourself a little bit of a buffer if that's what you're looking at. The only limitation with this drawing it out is 100,000 acre size limit. You can do a whole county as an AOI, and it will, but it's kind of one extreme or the other. 100,000 acres by drawing out or countywide. So just kind of a heads up on that. Otherwise, if it's more than 100,000 acres, you would just need to do multiple ones. But anyway, so moving on from here, then you can look at the soil map, and your area will have all the soils lines with each map unit symbol within there. And over here, you have the map unit legend, and it tells you the acres of each map unit. And if you click on one of these hyperlinks, it will open a new window with the map unit description. And so it's got everything about the area, the map unit, what are the dominant soils, the minor component, all kinds of stuff through here. This is automatically added to your soils report at the end. By default, you can not have it included if you want. I'll show you that in a little bit. But so next you go to Soil Data Explorer. So, and it automatically takes you to this tab first, but we're going to actually start over here on the ecological sites tab, because a lot of people come to Web Soil Survey to get ecological sites for whatever area they're looking at. And this is kind of, I'm going to take you from best way to get it to not quite as good, because there's like 3 or 4 options within here. So if you go to ecological sites tab, if you keep both boxes selected, you'll have the map and the component table. If you just want one or the other, just deselect whichever box you don't want. So when you select view all ecological sites, then it shows you everything within your area of interest. And below here, it has a table with each map unit and then each component, and then ecological site for each. Because like, especially like for this bracket low map unit, these 2 components have the same ecological site, but this other minor component is different. And so it's important to, have that, where some of the other reports, they just give you the dominant one. So it would just be this one. So from here, first thing you want to do is add to shopping cart. And that just will add it to your final soils report that you can save at the end. So you can say, okay, you can add a subtitle, whatever, if you want. But typically it's your own look, your own property or whatever. So you kind of already know where you're at. Okay, so from here, to get to the ecological site description, you can just click the hyperlink and it will open a new tab in Edit, which is like the Web Soil Survey of ecological sites. This is where they house everything. It automatically takes you to this page for the Southern Clay Loam. And this is kind of general overview is the first page. And if you scroll to the bottom is where you can download the full description. I would suggest doing this and saving them separately. And then you can have the ecological site descriptions and your report from Web Soil Survey saved or printed, or however you like to keep your, or both, however you like to keep track of things. From here, you can, if you save the whole report, then you'll have it as a PDF, but you can also, they have it broke down so you can view the report on the website. They also have print options. You could just print the sections that you wanted if you didn't want the entire report. And then if you don't want to click through the tabs and you hit all sections, it will just have the whole report on one big page, and you'll just scroll and scroll and scroll forever to get to the bottom. But I'm not going to spend a lot of time on this as far as like what's all in here. You can explore for yourself. Also, if you want to go straight to this website, if you just search edit NRCS or edit New Mexico State, it should come up within the first. search options. So that would be my suggestion as far as getting everything within that area. The problem with adding this to the only limitation, should say, not problem, on adding this to the shopping cart is that it will be in your final report, but these hyperlinks won't work. So you're going to want to do it while you're in Web Soul Survey. Okay, so other options to get ecological sites are back to the suitabilities and limitations for use tab. If you click on land classifications, you get a drop down of a bunch of different stuff. So if you use this ecological classification ID, it will just create a map with the ID numbers, which, don't always tell you a lot. You can also then do just ecological classification name. So if you hit view rating, it creates similar, color coded map. And then on the table below, it's just giving you the dominant ecological site that occurs. So like on the other one where you had multiples for including minor components, this just gives you the dominant one, and it also doesn't hyperlink. So you'd have to go to edit and search it for yourself. If you're not sure on any of these, what it's talking about, like what is conservation tree shrub group, just do the drop down and hit view description, and there's a little write up about what each interpretation does. Sometimes the title of the interpretation sounds more or less specific than it actually is. And so if you're not sure, it's good to kind of get an idea of what's in there. Let's see. So some other stuff within this suitabilities and limitations use tab that could be handy. Under land management, there's some more rangeland focused interpretations. All of these down here. If they have a TX after them, then it means that when they were created, the only parameters, like they were made for soils in Texas. So it's more strict for just Texas stuff, where a good example is these fencing interpretations. So if you do fencing, this is the national one. So they had to take in all the soils of the whole United States and kind of get some parameters from there. So if you do this rating, it's going to say that a lot of stuff is very limited. Where if you do the Texas one, they don't, like vertisols, they're not weighed quite so heavily. So you have a little bit, it gives you a less strict results, just because, I mean, we know that it's blackland soils, and you are kind of more aware of what's going on. So anyway, say you want to add this to your shopping cart, you just add there, and then it's got the rating, what the component, which component it rated, and then the reasons why it got the rating that it did. So this is basically just saying this Austin clay is somewhat limited because it has a high amount of clay and it shrinks and swells a lot. So let's see, what are some other good ones? So there's water, these down here, water erosion potential and wind erosion potential. Those are pretty good. I like to use those quite a bit. Underwater management is one for ponds, pond reservoir areas. That's a pretty good one. And one that's used quite often. Let's see. So if you're wanting to look more at specific soil properties related things, go to the soil properties and qualities tab. And it's got it broke down into these 6 categories. So if you're curious about like soil pH, that would be here. So this is one where when you do the drop down, it automatically opens advanced options below. Since pH typically changes with depth through the soil profile, you have to tell Web Soil Survey, what do you want it to pull the data from? So do you want it to be just the surface layer? You can click here. Or if you want a certain depth range, then you can enter it here. You know, depending on what the surface layer is, it may be 5 inches, it may be 10 inches, depending on what the soil is. Where if you put a depth, it will do a weighted average, regardless of, you know, where the horizon breaks are. So, and then you can also select different options in here. This is related to which soil component it's looking at. Dominant component will be whichever has the highest percentage within that map unit. Dominant condition is it will group, if some of the minor components are similar enough to the dominant component, then it will group it into there. So you kind of get potentially get more variation within the map unit. You're capturing more of that when you select dominant condition. A lot of the time, whatever the default is pretty good to go with. And you can kind of play with it and compare. So we'll just do dominant component surface layer for this one. And something else to keep in mind is when you get the rating here, this is the average for that component in like a natural setting. You haven't done any amendments, you haven't done anything to it. It's just kind of like the general kind of average of what the soil is. So of course, any kind of management practices and that kind of stuff could potentially affect this. This is just giving you an idea of like a starting point of where it would be at. It's not going to be a replacement for like a soil test or anything. So you can add this to your shopping cart. Some other stuff that is handy in here, soil erosion factors. If you know what K factor, T factor, if you know what that is, then you probably use it. If you don't worry about it, but this is where that's at. Soil health properties. They've just taken so like here's pH again. They've just taken all the soil properties and that are related to soil health and put them in one spot. So you could run all of those and add them to your shopping cart if you wanted. But again, so like with organic matter, it's going to be the average of what's just naturally occurring. Again, you haven't done anything to it, so it may be better or worse than what this is showing. Something else is soil qualities and features would be depth to bedrock or depth to restrictive layer. That could be potentially good to know. Drainage class maybe. And then water features is you got depth to water table, flooding, ponding. And so say you want these three water feature interpretations, but you don't necessarily, so like when you do it, when you run each one, it's going to create a new table and a new map for each one. Well, if you kind of already have an idea of where everything is, because the basic soil map is included in the final soil report already, and it gives you the map unit symbol and the map unit name, you can go to soil reports. And this just takes the mapping part out. So if you don't want the colored map, you just want the data in a table, then this is where you go for that. And so say you want those three water features from the other section, you can just go here and do view soil report. Oh, it's been so fast until now. And sometimes it's a little slower just because it's like it's combining more stuff. Okay, so you still have your soil map up here, so you know where those soil map units are occurring. But it's taken all that data and put it into this single table. So you've got and included even more hydrologic group, surface runoff, water table depths, ponding, flooding. And so although it's not colored here, you can still see like this bosky clay loam is occasionally flooded and its map unit symbol is BOA. So you would know that like here's a BOA map unit right here. So that's where that's occurring. And you can add that also to your shopping cart. Let me see. Okay, so say you've gone through everything and you've added all the interpretations and all the reports that you want to see. You come over here to shopping cart and this is kind of the last place to double check that your soil report is going to include everything that you want in here. It default selects everything except for glossary. So say you get to this point and if there's anything on this site you don't want, you just remove the check mark. If you get to this point and you realize, oh dang, I forgot to add whichever other interpretation, it's no big deal to just go back to that tab and add it and then come back to here. It's not, you can always go back and add more stuff. You can add another subtitle. You can change how you want it displayed. And then you click checkout. So if it's depending on the size of the area and the size of the report. So if you have tons of interpretations, you're only going to have the download later option, which you just enter your email. And then once the report is finished, the PDF is emailed to you. On the get it now option, it just generates it now. Sometimes it's a little slow, so I'm not going to show you that, but I have one that's already. So the final report will look something like this. So it will say custom sold report for Bell County. If you added the subtitle, it would be down here. And then it's got everything that you just looked at, but in a nice PDF form. So you've got the soil map, you've got the legend of all the map units that are occurring in that area with acreages for each, map unit description for each one. And then the exciting part is your interpretation. So when you add it to your report, you get the description already that's above it. And then below is the map with how everything rated. It gives you the legend, so you know exactly what each color means, and then breakout by map unit with the rating and then the reasons. So let me show you what the ecological site map looks like. So this was
Got about 1 minute, Ashley.
okay. So this was that first one that we looked at. So it just took the hyperlinks off. So that's why you want to print everything from edit first. The only other thing I want to hit on is this download soils data option. You can download your whatever the AOI is, and it makes a Esri shape file. So if you have an either ArcMap or ArcMat or ArcPro or any Esri product, you can use it in there. Not everyone has that. So if you want to download soils data, just contact me directly. There's a bunch of other download options that NRCS has now for soils data. And I'd be happy to help you get exactly what you need that can work for you, because it can be kind of confusing and stuff. So, and especially dependent on what, program you're using.
Thank you, Ashley. That was great.
Next up, we have Dr. Vanessa Korher Olson to speak on soil test analysis interpretation. Dr. Korher Olson was born and raised in China Grove, North Carolina. He's probably never heard a Doobie Brothers reference because of that. But she received her bachelor's in zoology and genetics from North Carolina State University in Raleigh. She received her master's and PhD in ruminant nutrition from the University of Georgia in Athens and Tifton, Georgia. And Dr. Korher Olson has been the Forage Extension Specialist at the Texas A&M AgriLife Research and Extension Center in Overton, Texas since 2008. Dr. Olson, I'll turn it over to you.
All right. Thank you, guys. Thanks for letting me join your party. So my topic today is interpretation of a soil analysis. So I will be happy to answer your questions in the Q&A box afterwards or throughout the rest of the presentations. So first and foremost, just as a quick reminder, how often you should be collecting soil samples. It will definitely depend on your management system. So as a forage specialist, I deal primarily with producers that are using their forages for grazing or for hay production. So if you are grazing, whether it's native range, improved or introduced forages such as Bermuda grass, like we have here in East Texas, if you're grazing, I recommend every 3 to 5 years. Since our livestock are recycling those nutrients, ruminants only keep about 10 to 30% of the nutrients they ingest. So they're recycling 70 to 90% of those nutrients back into that soil environment. So we don't have a lot of big changes as far as nutrients within that soil environment in a grazing system. In a hay harvesting situation, since we are removing a majority of the nutrients in those bales of hay, I recommend an annual testing or sampling because we're making big shifts in our soil nutrient status. And then we also have greater expectations as far as production level on those hay meadows as opposed to a grazing system. If you are collecting samples for other scenarios, whether that's turf, landscape, or gardening, those I would defer to someone else. Likely on a gardening situation, you may need to be doing that on a regular basis. However, most of what I'm going to talk about is in regards to forage production. So a soil test is a great way to know what nutrients are available and how you might need to amend those or improve those in order to support your management system, whatever that might be. So a routine soil analysis out of most of our university labs, routine analysis is typically at a 6-inch depth. It's how deep we should collect those soil samples. If we have bigger issues or concerns, or if we're growing something like alfalfa, we are likely going to need a deeper soil sample to look at soil nutrients deeper in that soil profile. So very quickly, a soil scientist once told me, if you send a soil lab a crappy sample, you're going to get a crappy analysis. So be mindful of how you are collecting those samples so you are giving them the best information so that they can also there in return give you the best data and recommendations based on your property and what you are trying to achieve. So sampling or analysis should be based on a composite sample. So that is multiple samples that are used to represent a given management area. And to us, a management area is no more than 50 acres. It could be 1 acre, it could be 20 acres, but less than 50 acres. So if you have a large acreage area, say you have 250 acres, that means that you're going to send more than a single sample to represent that 250 acres. So you're going to collect random samples at a 6-inch depth for a routine soil analysis, 12 to 15 cores at a 6-inch depth, randomly selected across that management area. You're going to mix those in a clean plastic bucket so you don't have any contamination from other maybe fertilizers or other chemicals that you might be toting around your property. You can fill that sample bag and then mail that immediately to the lab. If you are looking for soil test kits, such as the bags and paperwork for the College Station Soil Forage Water Testing Lab, those can be picked up at your local county extension office. Here's a picture of the bag. You do not have to use that bag, but we do need to make sure that we're sending at least 1 pint of soil for that analysis. And here's the website for the Department of Soil and Crop Science, our AgriLife Soil, Water, and Forage Testing Lab in College Station. Unfortunately, none of our other research and extension centers, like where I'm located in Overton, we do not perform any type of analysis for soil, water, or forage. So all of those samples do need to go to College Station. Also be mindful if you are working with a fertilizer retailer or a feed and seed store, they may do some sort of soil campaign or cover that cost for you, but know that they often or they may send your sample to a commercial lab. So what I'm going to talk about and show you today is for the Texas A&M lab. I'm happy to help you if you have an analysis from another lab to help you walk through and try to interpret that information, but be advised it will look very different than what we're going to see today in my presentation. So this is kind of a condensed version of what an analysis will look like. And this is a routine soil analysis. The crop grown in this scenario is an improved hybrid Bermuda grass that might be coastal or tipped in 85. It's not irrigated, and they're anticipating 3 hay harvests. So that is important to know as you are filling out the paperwork with your soil sample, that you tell them what crop you are growing, and whether it's being managed for hay production or pasture, or if it's both, that is obviously an option as well. They're going to use that information to give you the best recommendations based on your soil analysis. So the first thing that we're going to look at on this analysis is soil pH. So a routine soil analysis will tell you your soil pH, your primary nutrients, what is available, and then recommendations based on what you are trying to achieve, as well as what you are growing. So our primary nutrients are nitrogen, phosphorus, potassium. We'll also include secondary nutrients, calcium, magnesium, and sulfur. There are additional nutrients and add-ons that you can add to a soil analysis from College Station, but I'm going to focus on a routine analysis. It is important to know that at the bottom of your analysis in small print, there are some additional details, and it's critical to read those as well. There may be recommendations on timing of nutrient application, and other recommendations that are critical to be successful with your fertilizer application. So we're going to start with soil pH. So very quickly, soil pH is a measure of the hydrogen ion activity in the soil solution. Our forages can be very specific in regards to their tolerance of various soil pH. There are a lot of factors that influence our soil pH, and one of those is their parent material. So if we look at a soils map of the state of Texas, we have a lot of diversity. And so that supports our diverse vegetation across the state. So for me in East Texas and the piney woods of East Texas, we have very sandy soils. Sand is naturally acidic. No matter how we manage it, it naturally has a lower soil pH, well below that neutral level of 7. A lot of our soil pHs may be down in the 4s and 5s. Anything above a 7 is considered basic. Anything below that neutral 7 is considered acidic. Our fertilizer applications, specifically our nitrogen source, can have an impact on our soil acidity. So there are a lot of factors that influence that, but the only way to know what your soil pH is and whether or not that needs to be amended is with a soil analysis. We amend soil pH with lime applications. And if we look at that soil analysis again, based on this soil pH and this particular crop, there is a limestone recommendation in regards to trying to improve that soil pH. It is important to understand that our soils naturally change in soil pH from season to season, plus or minus a point throughout that time. So I always recommend collecting your soil samples at the same time of year, from year to year or every couple of years. So the fall is a great time for our warm season perennial forages. We do tend to have some moisture, at least in parts of our state, that can help make collecting those soil samples easier. Soil pH is critical because it can influence, depending on our vegetation or forage species, it can influence that plant's ability to utilize some of our primary nutrients. So moving on to the other parts of our soil analysis, our primary nutrients. One thing that's very frustrating for a lot of folks is that our units, so nutrients are listed in parts per million. So based on what's available, that's showing what's available within our soil environment. So in this particular scenario, we have 21 parts per million of nitrate nitrogen, and that's the form of nitrogen that's plant readily available. So a lot of people are like, well, I don't understand parts per million. The recommendation is in pounds per acre. That's, how I make my application. There are some quick conversions, some easy conversions. So if you multiply that parts per million by 2 and a 6-inch sample, that will give you pounds per acre. So if we look at that soil analysis again, 21 parts per million times 2 means there's 42 pounds per acre of nitrogen. And if we know to produce a ton of Bermuda grass, we need at least 50 pounds of nitrogen. So obviously we need some additional nitrogen to promote the growth of that hybrid Bermuda grass. So those are some easy conversions. You could take a quick screenshot of that, or I'm happy to share that additional information later if you have interest, but that's a quick, easy way to convert those nutrients. So like I mentioned, our primary nutrients, nitrogen, phosphorus, and potassium, are provided. Secondary nutrients, calcium, magnesium, and sulfur. If you have interest in micronutrients or have other concerns such as boron, what have you, those would be additional analysis. Do keep in mind, I mentioned earlier the use of commercial labs. A lot of our commercial labs do not provide nitrate nitrogen on a soil analysis report. They will provide other nutrients and oftentimes more information than College Station does. A lot of times those commercial labs will provide organic matter or cation exchange capacity. Those can be helpful in making fertilizer decisions. So keep that in mind as you look at using other labs. I'm always happy to help with interpretation of commercial lab results. Oftentimes they will not give a lot of recommendations, especially around nitrogen, because those commercial labs are historically used by our big crop guys that are growing cotton or corn. They know how much nitrogen they need to apply to support those larger crops. Very quickly, as I wrap up, I do have a website, foragefacts.tamu.edu. Under the publications tab is a likely helpful Excel spreadsheet. It is a fertilizer cost comparison Excel spreadsheet that you can download for free. This is a great tool. Now, keep in mind, this is primarily focused on forage production, whether it's grazing or hay production, but it's a great way to put in what your soil test is recommending and matching that with either a pre-made blend or figuring out what blend of nutrients are needed and what the nutrient ratios should be, how many tons or pounds per acre should be applied based on your number of acres. So this is a free resource. I'm happy to help you with this, to walk you through this, to put in your information and help glean additional information from this resource.
Thank you, Dr. Olson. That was great. Very pertinent for those of us involved in restoration or growing a crop or dealing with soils that may have been impacted on different things. We're going to hear from her again in just a few minutes, but next up, we're moving on to soil microbes. And so I'd like to introduce Liz Haney. Liz is a soil and ecosystem scientist with experience in soil testing and analyses, carbon and conservation practice modeling, and is the co-developer of the Haney Soil Health Test. Liz's purpose, drive, and passion are helping to improve producer profitability, environmental sustainability, and human health through regenerative practices and soil health. Throughout her career, she's developed an expansive network of scientists, thought leaders, consultants, farmers, and ranchers within the sustainable agriculture space. Liz loves nothing more than creating community and is skilled at coordinating and conducting educational events, workshops, and conferences, bringing together today's leading innovators in regenerative agriculture. Liz, I'm going to turn it over to you for our next presentation.
Hi, thank you for having me. So I'm going to talk a little bit about microbes in the rhizosphere. The rhizosphere is where everything magical happens in the soil. So when we look at soils, we want to understand and quantify the biological interactions that happen between plants and the soil and within the bulk soil as well. There are many tests now that integrate physical, chemical, and biological aspects of soil health and soil fertility, and more specifically, interactions in the root zone. So soil microbes, what they do in the soil, they mineralize soil organic matter and are responsible for nutrient cycling. They improve soil structure by creating organic matter glues that hold the soil together. They interact with other soil microbes in a synergistic way. They produce different biochemicals such as plant hormones and enzymes, especially like for microbial recruitment and controlling pathogens. So microbial recruitment. Microbial recruitment is the ability of a plant through its roots and root exudates, and specifically around the root hairs, to attract, select, and activate specific microbes from the surrounding soil community to fulfill nutrient needs, to provide stress resistance, disease suppression, and many, many other functions. So I'm going to give some examples of what we see right around the root zone. We went out and took soil from sunflower roots. So we pulled plants up, shook the soil off into a bag, right around that root zone and analyzed it. We also took some sorghum plant roots. If you note that the sorghum plant roots don't have as many fine hairs, the mass isn't as large as this on the sunflower roots. And we compared the two. Now, some of these measurements you might not be familiar with, but just wanted to show the difference between what we see in the rhizosphere of different plants and the bulk soil. So the bulk soil would be where no plant is growing. So you can imagine that's in between rows or a bare spot anywhere in your field. So 1 day CO2 is microbial respiration. That shows how active they are. Where we have a larger root mass, we can see that the microbes are more active under this sunflower versus a sorghum plant versus your bulk soil where no plant is growing. Their food source, which is water extractable organic carbon, is also more prevalent around that root zone than in the bulk soil. And basically the soil health score, if anybody has questions about that later, you can message me. But it's improved around that root mass. So what we see is that there is more organic matter, there's more interaction, there's more microbial activity around that root zone. It also shows up when we are looking at soils from the depth, from the surface down to maybe where there's anaerobic conditions deeper in the soil. So on this bar chart on the right, this is the surface, which matches up here with this picture of the soil profile, going down to 12 inches. Where there is more roots and more air and water interface with the surface, we have higher microbial activity, which is expressed by how large this bar is right here. We have more food source for the microbes. So up there where the plants are interacting with the soil, that's where the action is happening. And these guys right here that are breathing, they're responsible for nutrient cycling around that root zone and providing nutrients to the plant. We see that there is more organic nitrogen in the surface of the soil, more available phosphorus in the surface of the soil. So all of these interactions are happening in that critical root zone area. There's many different ways to measure microbes in the soil. Some of them are very specific, some are not. As I mentioned, microbial activity before, that's soil respiration. So microbes take in oxygen and give off CO2 just like we do. So the more microbes, the larger the microbial biomass, or the more active they are, you'll have higher soil respiration. It shows the overall health of the soil, and it's an inexpensive test, but it's non-specific. So we can't see whether it's bacterially dominated or fungal dominated, but it does give us an idea of the health of the soil. There are molecular ways to look at the microbes in the soil. For example, the phospholipid fatty acid test, and I'll talk about that in a minute. The advantages of that are it's relatively cheap, it's relatively fast. It does give us a quantification of bacterial to fungal ratios, but it's not super specific, and it is somewhat technical and kind of requires an in-depth knowledge of these different microbial communities in the soil. And then we can go even deeper into metagenomics, where we are identifying nitrifying bacteria, we are identifying arbuscular mycorrhizal fungi, we're identifying the different components and the functionality, as well as the diversity of these different microbes in the soil. It's very expensive, it's slow, and it's technical, but when we are doing research and really trying to figure out how that plant is interacting with the microbes in the soil, these genomic studies can be really a big advantage. So for microbial biomass and activity, the way we measure it is we take soil and we re-wet it to field capacity naturally through capillary action. And we measure the amount of carbon dioxide that the microbes give off within 24 hours. This microbial activity is directly related to nutrient cycling and especially nitrogen availability in the soil. And it's a reflection of how happy the microbes are in the soil. If the microbes are active, we know that we have good soil aggregation because they're getting air, because they need air to live and reproduce. And we know that they're getting good water infiltration because they need water to live. They live in a subaquatic environment. There are rankings for carbon dioxide. If anybody's interested in doing a CO2 burst test, you can do it on its own as a standout test, but it's also included in the Haney test. And we have rankings that show if there's very low respiration, it might mean that you're not going to have nutrient cycling. You're going to have really slow residue decomposition. If you have very, very high microbial activity, residue might decompose very quickly. We might be chewing up a lot of carbon, but generally it indicates a healthy soil. Keeping in mind, it's all climate and context dependent. What happens, how much CO2 respiration we have in West Texas in the arid areas is going to be completely different than, say, our Blackland prairies. So it's all context dependent. Another way we measure the microbes in the soil is we do a phospholipid fatty acid analysis. So all microbes have phospholipid fatty acids, and we can measure the total microbial living biomass as well as somewhat the community structure. So we pay special attention to our buscular mycorrhizal fungi. Our buscular mycorrhizal fungi are very important in the soil. They can well over double your root mass. They attach to the root mass. They are critical for phosphorus uptake and solubilization. We look at saprophytic fungi, which helps with residue breakdown. And we look at protozoa. So protozoa are little, tiny little predators in the soil that are mostly responsible for releasing nutrients like nitrogen because they eat bacteria that have nitrogen within their cells. In addition to everything being climate and context specific, microbial biomass changes over the year. And so she was saying, you know, sampling at a different time of year, you're going to get different results. Even with your standard soil testing, you're definitely going to get different results with biological testing because biology changes with the climate. It changes with rainfall. So this graph is showing this first number one here is the week of the year. So that's January. It's very cold outside. Microbes like a moderate temperature environment. Usually, generally, they're more active above 50 degrees Fahrenheit in the soil. But here it's cold. So the microbial biomass is actually much smaller. As it warms up in the spring, and you get some rainfall, the microbial biomass is going to start building. And as your plants start growing, let's say you have a corn crop you planted or something, and the roots start putting out food for the microbes, you'll get spikes in microbial biomass. So their biomass will build in response to those plants being in the ground. That's why in regenerative agriculture, especially, we talk about keeping a living root in the ground at all times. And that's one, because they feed the microbial biomass. Microbial biomass is responsible for carbon storage, nutrient cycling, all of the wonderful things in the soil that happen to feed your crops. Also, plants, a living root, takes in carbon dioxide from the air and pumps down about half of that, which it takes in, to the roots and to feed the microbes. And so we see a big bump when there's a living plant on the ground growing and then dropping off as it gets colder in the plant, and you might be in a fallow situation. It gets colder, the microbial biomass dips down. So plants release chemical signals, which include sugars, different acids, amino acids, enzymes into the rhizosphere, and they attract microbial partners. So there's biological matchmaking, and it's targeted to the different types of plants and responsive to at what plant stage the plant is at and what it needs. So if a plant needs phosphorus at a specific time, it can recruit microbes that solubilize phosphorus. This is an example of that. These are 15 plots that we had showing wheat growing. Everything was treated exactly the same. So microbes were actively recruiting phosphorus when the plant needed it. Another example of this is different plants like buckwheat. They feed rock-eating bacteria. They recruit them and they increase available phosphorus by double in this example. I'm going to skip over some things. One I want to show is that root hairs actively take in bacteria and then they push them out. And this is called rhizophagy. So this is a complete another step at looking into the soil microscopically and the plant root interactions. If anybody has any questions, this is my contact information.
Thank you so much, Liz. That was fantastic. All right, so we're ready to get our last presentation started. We're ready for Dr. Vanessa Corrier-Olsen to come back and share with us about alternative fertilizers, the good, the bad, and the ugly. I know we all hear of different things that could be used to fertilize the soils, and so I'm excited to learn more about the science behind these choices. So I'll turn it over to you, Dr. Corrier-Olsen.
All right, thanks. So alternative fertilizers, of course, we have a lot of options often in regards to sources of applying the nutrients we need for vegetation or forage growth. And so some of those can be good, bad, and not so good. And so I will forewarn you that typically when commercial fertilizers prices go up, which they've already been going up this spring and summer, early part of the summer, and we're anticipating some more price increases. We tend to get hit with a lot more of what I like to refer to as non-traditional products. Sometimes I also like to refer to them as snake oils. Something I was taught growing up, and I'm still learning, of course, is if something sounds too good to be true, it often is too good to be true. So we're going to talk about what some of these alternatives might be, some positives, negatives, what you might actually get out of some of these products, and whether or not they're actually going to provide the nutrients you're looking for. So this is a kind of a table of non-conventional soil additives. And so that is very important. There are products that are additives and they are not labeled or not categorized as fertilizers. So fertilizers are going to provide nutrients like nitrogen, phosphorus, potassium, magnesium, calcium, sulfur, what have you. But there are a lot of other products that if we actually read the label, like we should, of any product that we're using, whether it's a pesticide or a soil additive or soil amendment, if we read the label, they may not actually be supplying any type of nutrient, which might be our desire or what we're looking to do. And so we're going to talk about some of these. So there are animal byproducts. Some of them, as we'll talk about in a minute, actually have some benefit. You may come across a fish emulsion or a fish meal. So a very common as far as an alternative source of nutrients. They do consist of nitrogen, phosphorus, and very little potassium. So the amount of those nutrients or the ratio of those nutrients is going to be based on how that product was digested or prepared. I can imagine, I've never worked with these, but I can imagine there might be an odor that may or may not be pleasant to you and your neighbors. But keep in mind, so if we look at applying this product to a very large acreage, it can become very uneconomical based on if we're trying to meet a higher rate of those nutrients with a very low ratioed product. Oftentimes these products are going to come in smaller quantities, maybe in a gallon, 2.5 gallon jug. And if we do some math, some pen to paper and a calculator, we'll learn very quickly it becomes very uneconomical to use some of these products on large acreage. This might be great for a home garden or lawn scenario, especially if you're trying to be organic in your particular scenario. There are other manure and compost-based products. You see these at Lowe's and Home Depot all the time in 25 or 50-pound bags. They do consist of nitrogen, phosphorus, and potassium. In these smaller quantities, however, they're going to be uneconomical for a larger area. So a 250-acre pasture, that's not a way we want to apply any type of animal manure. Isn't a product that's sold at Lowe's or Home Depot that's obviously more directed to a home or a land homeowner or a small landowner, as opposed to many of us on a farm or a ranch. We do have animal manures that come in larger quantities that we can utilize that can provide nitrogen, phosphorus, potassium, as well as some other benefits as well. A lot of our animal manures, depending on our location in the state, is going to impact what may or may not be available to us. Obviously, if you're in the Panhandle, there's quite a few dairies. If you are in parts of Central and East Texas, we have some poultry litter that could be broiler, layered litter that can be available to us. Keep in mind, if commercial traditional fertilizer sources go up in price because of the demand on these animal manures, their price will also go up. This table shows us that it's highly variable. The nutrients within that product are going to vary from farm to farm and in a poultry scenario from house to house, even on the same property. So the numbers outside of the parentheses show an average in regards to how many pounds of nitrogen, phosphorus, and potassium might be in that product per ton. And the numbers within the parentheses are the range. So highly variable. If you have questions about those, I'm always happy to answer. There are a lot of pros and cons. Obviously, with any type of animal manure, there are going to be odors that may or may not be offensive to you and your or your neighbors. High variability. You can have these manures tested at the College Station Soil Forage Water Testing Lab, and they will tell you how many pounds of nutrients are in a ton of that specific product that you have provided to them. So this can be a great alternative, but we do need to pay attention. If we are using animal manures, we need to soil test on a regular basis because we can oftentimes be over applying nutrients, especially in the case of poultry litter. We're often applying way more phosphorus than any of our forage crops might actually need. So we need to be environmentally mindful of that. So we need to be soil testing on a regular basis. Humates or humic acid, this is one that I've had questions about before. A previous retired soil scientist, Mark McFarland, out of College Station, did some evaluation on humic acid in corn to look at if it had an impact on corn production on yield. And it did not make a difference when added or used alone with or without traditional fertilizer sources. So it's usually made from coal or clay. It contains a high percentage of humic acids and organic matter. It requires 2 tons of humate per acre to increase our soil organic matter by 0.1%. Most people are using humate for that purpose to try to increase organic matter. It is very difficult. It takes a lot to increase organic matter in our soils, and it can take a very long time. So this is a product that you may come across, has not shown a direct impact on yield of any type of particular crop. Seaweed products, there's a C90 mineral. This is apparently a Neptune's harvest. They're typically made from kelp that's been harvested, dried, and ground. Soil application rates are fairly high for forages. Very little nitrogen, a trace of phosphorus, and a small percentage of calcium with some additional micronutrients as well might be supplied. But once again, this is a product that would be needed in large quantities to have any impact on a forage system, especially in large acreage. So our previous speaker was talking about microbes. So that's something that I get a lot of questions about, and you see products that claim to add microorganisms to your soil environment. There are some microbial inoculants. Rhizobium is an inoculant that we utilize in legumes, and I would consider legumes an excellent alternative. It's a way to recycle nitrogen back into our soils, especially if we are grazing those legumes, whether they're cool season or warm season. The best way in a forage system is through grazing. So we're taking advantage of that higher quality forage, and then we're recycling that nitrogen back into that soil environment. Please know that most of our, especially our cool season legume use, is primarily east of I-35, where we have higher average annual rainfall. There may be some native legumes that we can utilize in our more arid environments or under irrigation in other parts of the state. Do keep in mind they can be sensitive to soil pH, and they will require phosphorus and potassium, but we can recycle that nitrogen. So that's an alternative. A lot of folks here in East Texas utilize cool season annual legumes for that purpose, to attract wildlife, to provide higher quality forage during that spring, winter feeding time, as well as to recycle that nitrogen in a grazing system. Free living soil organisms, so microbes. Like I mentioned, I get a lot of questions about adding microorganisms to our soil environment. A lot of times those products claim that by adding microorganisms, that makes nutrients that are already in our soils more readily available. At a soil scientist basically explained to me that if you have vegetation growing, whether it's a field of goat weeds, mesquite trees, Bermuda grass, native grasses, if you have actively growing vegetation, we do not necessarily need to add additional microorganisms into our soil. Our soil should have what it has. Now, when we might need to add additional microorganisms is if we have soil that has been neglected and abused and not even a lonely goat weed would grow on it, then we would need to likely add microorganisms to help rebuild that soil environment to support vegetation growth. Another time we might use microorganisms is to microbes will break down herbicides or pesticides out of our soil environment. And so they can be used to clean up a pesticide spill. So that's another scenario where we might need to add additional microorganisms, is to help break down those herbicides that were accidentally dumped or spilled in a soil environment, to reduce the impact that they might have in that location. Additional products, enzyme-based products, they are typically advertised to improve soil structure, to make nutrients more readily available, to improve drainage, water retention, all of the things that we're looking to do within our soil systems. These enzymes typically are broken down by microbial action. And so they're likely to perform best in soils that are already well balanced and in good condition. So they're not going to take a poor environment and make improvements. They're going to work better in an already high functioning soil environment. Soil conditioners are typically wetting agents and surfactants. So they improve water penetration. If, typically it's not something that we need to add to a soil environment. Wetting agents are not going to have any effect on poor drainage due to hard pans or any type of compaction. So if you have soil compaction, there are other mechanical ways that we can alleviate that to allow for deeper root penetration. Dr. John Jennings at the University of Arkansas has been able to do some evaluation of some of these products on Bermuda grass. In this particular table, we have our highest dry matter yield on Bermuda grass at the very top, going all the way down to the least productive. So those three products, when they were applied by themselves there at the bottom in the green, did not produce any more Bermuda grass than doing absolutely nothing, if we look at those numbers on a statistical basis. You will also know that notice if you're reading the label for these products, they will often tell you that they are meant to be used in addition to your traditional fertilizer practices. So in addition to urea, ammonium nitrate, potash, what have you. So a lot of these products are not meant to be a standalone product. So once again, I highly recommend making sure you are reading the labels for those products. Reach out to folks, your county extension agents, folks that you have heard today during our plant party. We're always happy to help research or look into a particular product. You can always do your own evaluation. If you're just curious, you want to try a particular product, but make sure you are doing kind of a small scale trial, that you are comparing it to doing absolutely nothing, or maybe to your previous practices. A lot of people will have 100 acres, they'll get a product, they'll apply it to that full 100 acres. And because they want to see an impact, and they have nothing to measure against other than their memory, they might actually think they've seen a difference. A lot of times with these products, you're applying a lot more water than actual any level of nutrients, and all of our forages or plants are going to respond to water. That is a critical nutrient for forage or plant production. So you can do an evaluation. It doesn't have to be at this scale. You can take 100 acres, split it up into multiple 25-acre tracts, whatever works for you, to compare different products, to compare to a control where you're doing nothing, and compare to what you have done in the past versus potentially a new product that has been marketed to you. So once again, use that pen, paper, and calculator and reading those labels to really determine if this product is going to provide the nutrients or what you need for plant production and growth. So once again, my website, foragefacts.tamu.edu. You can find my contact information on this website if you need assistance with reading a soil analysis, or if you come across a product you want someone to help kind of dig into, get more information, or find out if it's something that we've evaluated in our research here at Texas A&M. So thank you guys for letting me join your party.
All right. Thanks so much, Dr. Correlson. That was a great presentation. Really appreciate that. And thanks again to our door prize sponsors, Bamert Seed, InView Range and Pasture, Native American Seed Company, Quail Forever, Renewable Resources Extension Act, Texas A&M AgriLife Extension Service, and the USDA Natural Resources Conservation Service. We appreciate your support of the Plant Party. Thanks again to all our speakers and to all of you for attending our 19th Plant Party webinar. Again, Plant Party is a quarterly advanced plant webinar training series brought to you by USDA Natural Resources Conservation Service, Texas Parks and Wildlife Department, and Texas A&M AgriLife Extension Service. Our next Plant Party will be on September 17th with the theme of plant management before dormancy. You won't want to go into hibernation before that exciting webinar. We will send an email with more information to all past registrants. Until then, we'll see you next time.